everyone. I'm excited to share this presentation with you today, and we are going to cover flow cytometry basics, how you can overcome technical challenges. My name is Erin Christensen, and I'm a senior field application scientist with Miltenyi Biotec, and I cover the Southwest region for the United States. So today we're going to go over basic principles of flow cytometry, optimal panel design for data acquisition, and then how you can implement standardized workflows with your flow cytometry experiments. So first of all, what is flow cytometry? Well, cyto means the cell, and metry means measurement. So flow cytometry is the measurement of cells and their characteristics, function, or activity you want to look at as those cells flow through a liquid stream. So let's think about what is measured on or in a cell. And so we're going to first look at intrinsic physical properties of just the cell itself. And so here we have a laser, and my cell is going to come in contact with the laser, and there's nothing added to the cell right now. So we're looking just at scatter properties of the cell. And let me get my pointer options. And so in the front here of your flow cytometer, there is a detector, and it is known as forward scatter detector. So it is going to pick up this FSC signal. And with the forward scatter, we can determine relative cell size. And that's what you're seeing with these wavelengths here. This is our forward scatter, and the detector would be in front of it. So it's almost like a shadow of a cell. So the forward scatter is how we can tell relative cell size. Now there's also going to be a signal coming off of the side at a 90-degree angle, and that is our side scatter. And the side scatter is able to pick up internal complexity inside the cell. And quite often you'll see on flow cytometry plots, you'll see FSC plotted against SSC. And just using these two scatter phenomenons, we can take a look and see different cell sizes of populations. And that's what we're looking at on this particular plot. We have the forward scatter on the x-axis, we have side scatter on the y-axis, and we're running some human-lysed whole cell blood. And just scatter alone can see different cell populations. And this first population, this more dense population that's low in side scatter, it's a relatively small cell, and that's going to be our lymphocytes. Here we see a second population, slightly bigger in forward scatter and slightly bigger in side scatter, and that would be our monocyte population. And then this large diffuse population up here at the top, this would be our granulocytes, with a lot of different vacuoles inside of the cell being quite a bit higher, and we can see these differences just based on scatter alone. Well, quite often we want to look at more than just intrinsic physical properties. We want to look at extrinsic physical properties or functional properties, and we do that by adding fluorescence to a cell. And when we add fluorescence, we can look at different things like gene expression and intracellular proteins, metabolic activity, cell proliferation, lipid content, as well as surface proteins. Well, where are we going to get this fluorescence? Well, it's really all around us in nature. There's a lot of fluorescent rocks. There's fluorescent microorganisms. And you've all heard of the bioluminescence that's given off by algae in our oceans. And so harnessing the power of fluorescence, we can tell more things about a cell as we run it through our flow cytometer. Well, we first need to understand the principles of fluorescence. So this pink circle here, this is going to be my fluorescent molecule. And it's going to get excited by a high energy, which is going to be our blue light coming from a laser on our cytometer. So when this fluorescent molecule gets excited, those electrons are going to jump from their ground state up to this excited state. And this excited state is not very stable, and a little bit of that energy is going to be released with heat, but the majority of that energy is going to be released as at a longer wavelength and a lower energy before those electrons go back down to their ground state. And so this emission coming from this fluorescent molecule is what we're going to harness and we're going to use to detect our signal off of our cells. So we're going to look at excitation of the fluorescence and emission spectra for a couple different fluorochromes. And what I've got on the x-axis is our visible wavelength, where we span from 350 nanometers all the way down to 800 nanometers. And on the y-axis, we're going to look at our relative absorbance and emission. And the first dye we're going to look at is Vio Blue. And we can see the Vio Blue has an excitation profile that spans from 350 nanometers to 450 nanometers. You'll see that there's a peak excitation at the top of this little peak here, and this corresponds nicely to a violet laser that may have a excitation profile, the violet laser of 405 nanometers. Now, after the Vio Blue gets excited, the emission is going to be shifted down, and you will see the emission spectra is going to travel from 425 to 525. We can harness this emission and pick it up with our cytometer. Now, the next dye we'll talk about is Fitzy, which is a very common green fluorochrome that's used with flow cytometry. And Fitzy has a long excitation and also has this long tail. So Fitzy can be excited by a couple different wavelengths. After Fitzy is excited, the emission spectra, the emission is going to travel from 475, and you'll see the corresponding very long emission bleeding into multiple wavelengths. Now, lastly, we're going to look at APC, and this is a large protein, and it has a very long excitation profile. And then with the emission of APC, you'll see that emission is very close. The excitation and emission, the transfer, this is known as the Stokes shift. So longer wavelengths have a very short Stokes shift that you'll see with our red colors, and our more smaller wavelengths or higher energy will have a larger Stokes shift that you can see in the Vio Blue, for example. You'll see this is a longer distance in between the excitation and emission for violet colors. So what kind of fluorescent reagents are out there that we could use with our flow cytometer? Well, the first category I like to talk about is something we call fluorogenic probes, meaning they're naturally fluorescing, and they also have a high affinity to bind to what we want to look at on a cell. And Nile red is a common example that's used for determining lipid content of cells. CFSE has a really long name, and I'm not going to attempt to say the name, but most people just refer to it as CFSE, and it's used to label all proteins, and it's great for cell proliferation assays. And propydium iodide is a great reagent, and it labels double-stranded nucleic acids, and it's used for viability as well as DNA content analysis, such as cell cycle. And you can see how PI is going to go into this double helix here. Now, the next category is fluorescent proteins. It came from our first one, our green fluorescent protein, GFP, and we can track that protein expression as we watch the color into the cell, and we can track that GFP signal, and we can see that fluorescence of the protein expression. And there's every color of the rainbow of fluorescent proteins available now. So if you have a yellow fluorescent protein, or yellow is your favorite color, you can use YFP for your experiments. Now, lastly, these are components that bind really nicely to cells, but they're not naturally fluorescing, so we're going to conjugate it to a fluorochrome. And annexin is used quite frequently. It binds to phosphatidylserine, and it's tracked on apoptotic and necrotic cells. Soluble ligands will bind to various different receptors. And I think most everybody has heard of antibodies that will bind to various targets, and they are coupled to fluorochromes. So what is a flow cytometer? Well, it's a combined system that has fluidics that will move the particles through the light path. We have optics that are going to generate and detect the light signal, as well as electronics that will convert that optical signal to an electronic signal. Well, let's take a look at fluidics first. And fluidics just helps align and have the subsequent measurement of the cells done by the laser that we can see happening on each of our cells being lined up here. This particular image is showing the flow cell and our fluidics from our max quant system, and we use hydrodynamic focusing on our cytometers. And we have sheath fluid that goes through our cytometer. We have the narrowing effect here with our hydrodynamic focusing. And when we inject our cell sample, it travels through the middle of the sheath. The sheath brings the cells in our stream into the flow cell where they get interrogated by the laser, and we can see the signal being generated. Moving on to the optics, this is where, again, we have to have that excitation to whatever fluorochrome we've attached to our cell. We have the emission of that fluorescence, and then we're going to detect it in our PMTs. Our max quant cytometers have 3 different lasers in them. And so our cells are going to interact with our red laser first. They're going to emit the energy. We pick it up in our detectors and we see our pulse signals. Our cell travels through the blue laser and we see our signals coming off. They're emitted, we're detecting them, and we see the peak pulses. And then finally, our violet laser, we see our signals, and then everything, all these measurements are put together to see the events that we see on our cytometer. So let's talk about the lasers first. The laser is what is going to, again, excite our fluorochrome. And the output from that laser is going to be monochromatic, meaning it's only going to be one color at a time per laser. And that laser is going to be very bright. It's going to have that high intensity that's needed for excitation. And it's unidirectional, meaning it's always going to go into a one wavelength, one wave at a time. So only one directional. And you can see there's many different colors of lasers out there. We have UV lasers, violet, blue, green, yellow, red, many different lasers, but this is used one color at a time. So now that we've excited our fluorescence, we now want to pick up that signal. And we can do that by having some different filters. And what that does is it allows us to start going in and pulling out specific signals from that entire emission that we looked at earlier, because some of the emission passes cover several nanometers in size. So there's short pass filters, which allow the shorter wavelengths to travel through, and they block the longer wavelengths. The inverse happens with a long pass filter. It allows the longer wavelengths to pass through, blocking the shorter wavelengths. And probably the most common filters that are used in a flow cytometer are going to be bandpass filters here. And bandpass filters have usually a nomenclature of like a 525, a dash or a slash, and a 50. And what this means is the first number is going to be the middle of the filter. And then the second number is going to be how wide that filter is. And so this particular bandpass filter would pick up the signal from 500 nanometers to 550 in this particular example. There are other features inside an optical bench, such as dichroic mirrors, which can allow some filters and some wavelengths to travel through, and then also bouncing the light down another avenue of the optical bench to be picked up by other filters. So now that we've picked up a little bit of our emission, we want to talk about what voltage will do to your signal detection. And so your signal is picked up by a PMT or a photomultiplier tube. And just like it sounds, it multiplies the photons. The photons that are released from the emission of the signal, the photons hit the photocathode on my PMT pictured here. And then the photons will fly off electrons, and those electrons will travel between these diodes, amplifying the signal before having the electrical output. And that amplification of the PMT is adjustable by increasing or decreasing your voltage and gain on the detector. Let's see what it looks like on a particular plot we look at on our flow cytometer. So at 200 volts, our signal is down here in this lower left corner. As I increase that voltage, what it does is it moves that signal. After amplifying the signal in our PMT, we see the signal increase and it moves to the middle of the plot. Increasing the voltage even more will move it further to the right on our particular plot. Increasing using your voltage will move your population further to the right or up, depending on which channel you are looking at. So now that we're talking about our signal generation, how are we going to change that photon to volts again? What does it look like? Well, this is the voltage pulse that we were talking about. And in a visualization here, your cell starts to enter the laser. This is the full time in the laser, and then the cell exits the laser. And the voltage pulse is generated exactly like it looks like as that cell gets interrogated by the laser. And so there's different measurements we can do from this signal. We can take a look at the height, which is the maximum number of photons that were emitted during that cell interrogation with the laser. You could also look at the width, which is the time from the signal initiation to the signal completion. And you can also look at the area, and that's the reason that is the signal that most people take a look at, which is the full area and the full quantity of photons that are emitted over the full time as the cell interrogated with the laser. So now that we've generated a bunch of data, how are we going to display it? What does it look like? So we have display of data in a pictorial format, such as a histogram here. Well, all of the signals that are coming through my cytometer is just a big list of data. So imagine, if you will, this giant Excel spreadsheet with multiple channels. We're looking at height, we're looking at area, we're looking at all of the different channels of our detector, as well as the lasers. And each of those lasers is going to excite it. So we have a lot of data. So instead of trying to interpret all of this data in an Excel spreadsheet, what we can do is we can start plotting all of these individual pieces from this list of data. And for example, we can look at a histogram. And what we've done is we have a whole table of data that we have compiled, and we've looked at males and females and their weight, for example. And on the x-axis, we're saying in kilograms, how many kilograms does each individual weigh? And we can count the number of times we've seen this result on the y-axis. And so what we can see is we have females plotted in blue, and we have males plotted in this orange color or yellowish color. And again, think about that's all of those individual pieces of data, and we've just stacked them on top of one another in a histogram format. So if we think about the same type of phenomenon, what's happening at a fluorescence level on the cell, if we have our increasing fluorescence intensity on the x-axis and the number of events on the y-axis, we could think about, okay, down here on this lower left, maybe these are cells that don't have any of my fluorescence antibody attached to them. This lower signal here, this middle part, maybe these are some cells that have a little bit of the signal that has been bound to the cell. And on this far right, maybe these are cells that have that high abundance of my signal generation and my antibodies have bound to the cell. So that's an easy way to visualize a histogram of data. What happens and how do we get all of these dots that I've seen on flow cytometry plots in the past? Well, these are just two histograms stacked together. And instead of looking at one parameter at a time in a histogram, we take both of those events and we place a dot. So we see the first event where we have FSC of 30, and then we have SSC of 60, and we place maybe a dot in this green section, which is our lymphocytes again. Now we have the event 2, which is the signal, and it has a higher FSC of 100 and our side scatter of 160. So we go up 100, we come up 160, boom, we place a dot in this red area, and that's going to be an event for our monocytes. And then same thing for event 3. This has a larger forward scatter as well as a larger side scatter. So we go a little bit further up here and we come up and we place a dot in our granulocyte population. Well, what about looking at two fluorescence parameters? So if I place on the x-axis a signal for FITC and on the y-axis my signal for PE, what we could think about in this 2x2, down in this lower left, these would be cells that would have neither FITC or PE on them. And my population that's on this lower right, this would be all of the cells that would have just FITC by itself on it. The cells that are in the upper left quadrant would be cells that have just PE. And then in this upper right quadrant would be cells that would have both FITC and PE attached to them. However, this region could also contain cells that are singly PE only and singly FITC only, and then that could be stuck together like a doublet, and we would want to find a way to exclude these from our analysis, and that's for another gate later. Now, when we look at scatter signals, we've been looking at forward scatter and side scatter, and quite often with your preps and your samples that you're running on a flow cytometer, you get a lot of really small particles. And small particles are going to be in this lower left, really low on side scatter area. This could be debris, this could be red blood cells from your prep. And quite often you don't want to look at this little lower, these smaller particles. So you can set up and use what's known as a trigger or a threshold. And by using a trigger or threshold, we've excluded all of those small particles. And so it's a threshold that says to the cytometer, only keep events above this particular level, and then you will see our true white blood cells that we see in this forward scatter, side scatter. And this is very helpful for statistics because you can see in this other prep on the left, 73% was just debris or white blood cells that we wouldn't want to look at and analyze further. And here we have cut down that amount of debris to 6.5%, and then we can look at our true cells that we want to analyze, which are going to be our PBMCs. Now, there's different ways of looking at your data. We've been looking at black and white dot plots. But you can change instead of just looking at this big black hole here of data, we can change this to a density plot. And so you can see on the right, this density plot makes the data look quite a bit nicer. And I can see where my biggest abundance of cells is, kind of my hot zone in this little area here. And then I can see the fewer cells. So it just gives me better visualization of where my cell population is. There's other plot types available. There's histograms, there's contour plots, many different ways of visualizing your data instead of just looking at a big black hole like we see on the left here. So the power of flow cytometry is acquiring a bunch of cells very quickly. But then what do you do with all that data? Well, you can start pulling out and subpopulations, and you can do analysis. And so you can use gating, for example. So how would you do gating? I thought it would be fun to kind of do a different visual representation of gating. And so I have a group of people, and we're looking at their faces here. And so imagine, if you will, these are like your cells that you've run on your cytometer. So I want to look at all these human faces, and I want to determine how many of these people are wearing glasses. How many are males and how many are females and how many are wearing glasses? So I could do a gate. I could draw a gate around these different faces and say, I have pulled out the first population, P1, are people wearing glasses. And then I can draw another gate and I can separate them out into male versus female. And that's P2 and P3, my second population, my third population. And once you start pulling out your subset of all of your different populations after acquiring data very quickly, you can also get statistics on it. So now that we have talked about what a flow cytometer is, how we can harness the power of fluorescence, we want to talk about how we can design a panel for acquiring some data. So there's different categories we want to look at. We're going to first look at, we need to understand the capability of our instrument. And we're going to look at our max quant 16 that we have highlighted here. So when you choose a cytometer, you want to find out how many lasers are on that cytometer. And that's important because we learned we need that for excitation. What are their wavelengths? Because we want to pick the fluorochromes that will be excited by these lasers. Once we have the excitation, how many detector channels are available for each laser so we can pick up the emission of our different fluorochromes. Speaking of fluorochromes, which fluorochrome should we use and can be detected on each of the cytometer channels? So there's many different factors, but that is important. And now that we understand the basics of how fluorescence works with the excitation and emission, we can take a look at what lasers are available. And we can see 3 lasers are on my max quant 16, and that's necessary again for the excitation. The filters are going to be where we're going to be detecting that emission coming off of each fluorochrome. And you can see most of our filters here are those bandpass filters that we talked about. And so this particular V2 channel is going to pick up from 500 to 550 nanometers. We can see some example Miltenyi Biotec dyes as well as other vendor dyes that can be run on our max quant 16. So now that we have our cytometer, we understand what channels are available, let's pick out and look at how to design a staining panel. So what is an antibody panel? Well, it's a set of different antibodies that are coupled to different fluorochromes. I have a few T cell markers picked out here, and they have some colors attached to them. But are these the right antibodies to look at? Are these the right fluorochromes? Which ones to choose? It can be pretty complex if you don't know what you're doing. So as you're thinking about different antibodies to use for your panel, we need to talk about stain index or stain intensity. So what is the stain intensity and what do we need to think about for our staining? Well, we first need to think about the expression level of the cells that we want to run on our cytometer. How abundant are those markers on the cells? So we want to look at the expression level of the cell and we want to balance it with the brightness of the fluorochrome, and it is a balancing act. So if we think about stain index, this is how the formula has been done for the stain index. So we want to see a separation between a positive signal and the negative signal without oversaturating the negative population here. So this is the formula on how the stain index has been calculated. Don't worry, you don't have to calculate it. There are a lot of fluorochromes out there that have the brightness based on the stain index already calculated for you. And you can see here's some of our brighter fluorochromes. VioBright 515, APC, and PE are two very commonly used very bright fluorochromes. And then we have some of these we call kind of the middle of the road. So they're bright, but they're not really bright like APC and PE. And then we have some dimmer fluorochromes down here. And I have people saying, when would I ever want to use a dim fluorochrome? It's a balancing act. We are able to use these for certain markers. Now, if you're just starting out and you're like, I have no idea the brightness of a signal, there's a great website that I love to use, and it's called fluorofinder.com. And you can put in your cytometer that you want to use. And when you put in your cytometer, it'll give you the filters and it'll give you some suggested fluorochromes. And visually, you can already tell what's going to be a bright fluorochrome. As we mentioned, PE, I have 5 tick marks here. So I know this is a very bright fluorochrome. Here's my APC with a very bright fluorochrome. If you knew nothing about Pacific Blue, you can see it only has 1 tick mark, and you're going to assume, and I can see, that's probably going to be a dim fluorochrome. So let's start building a panel. And so when I build a panel, I like to break it down into 3 different categories. I want to think about primary antigens first. So this is a primary antigen, and it's going to be brightly expressed on the cell, meaning the cell is going to have a lot of abundant epitopes for my antibodies to bind to it. This particular marker, these antigens, they're used for gating. They're going to have an on or off pattern. So the cell is going to do the work for me. It's going to be very bright. I'm going to have a great separation. And some examples of different primary antigens are CD4 as visualized here. We have CD3, CD8, GLAST for an astrocyte marker. So since the cell has so many markers and it's very brightly expressed, I can use some of those dim fluorochromes. I can balance that out. So once I have my primary antigens picked out, I can move on to the second tier, which is going to be my secondary antigens. When I see the expression level, it's not going to have that huge separation like I have for a CD4, and they may have kind of a smeary effect. But I can still draw a gate around them like I can do here. I can gate them, I can pull out that population, and I can start doing my hierarchical gating. So if I can draw a gate around it, it's one of that middle of the road kind of marker. So I can use some of that middle of the road brightness, and I can do that FITSI or APC, for example, for some of my subset markers. Well, lastly, these are going to be my tertiary my antigens, meaning my tertiary antigens are very dimly expressed on the cell, or maybe it is a rare event that is in my total cell population. For an example, a rare event would be looking for a stem cell in a population of PBMCs running on my cytometer. So since it's rare or dimly expressed, this is when I want to pull out the big guns of my bright fluorochromes of my PE and my APC or my VioBright 515 to make sure I can see that signal. Then I can put it all together, and again, your bright antigen on the cell, put it with a dim fluorochrome. Kind of a medium brightness of the antigen, put it with a medium brightness of your fluorochrome. And those really rare or dim antigens pair it up and balance it out with a very bright fluorochrome. Now, I have some people tell me, I'm working on a new project. I have no idea if this is an abundant marker or a bright antigen or a rare antigen. So here's a hint. If you go to a vendor's website, and if you take a look at FOXP3, for example, and you're like, I don't know anything about FOXP3. And if you see FOXP3 is available only in a bright fluorochrome, such as PE and APC, it's only available in 2 colors. To me, that tells me that is a dim antigen. And I'm pairing it with a bright marker, a bright fluorochrome, so I can see it. As we move up the list and I see a marker, I see an antigen like a CD3, you see it's available in every color of fluorochrome here. So I can make an assumption that that's probably a very bright antigen on the cell, and I can put any color available with it. So when you are building a panel, my preference, I like to start off with my rarest or my few choice options, my dim antigens first, like a FOXP3, because I only have 2 choices available. And then I start picking out fluorochromes that will match to each of these different markers. And I use CD3 last because I can put any color in there after I've picked out fluorochromes for my other antibody choices. Other things to think about are some of our fluorochromes will spill into other fluorochromes. You saw me show you AP or FITSI that has notoriously a long tail that will spill into another adjacent fluorochrome. So what I could do is I could spread those fluorochromes between 2 different cell types. And a common spillover has vio-blue and vio-green. And so vio-blue and vio-green spill into each other like we're seeing up here. And so I can put vio-blue on a B cell marker like CD19, and I can put vio-green on a T cell marker like CD3, for example. And then those 2 markers and those 2 fluorochromes will not be on the same cell at the same time because they're on different unique cells. All right, so we know what our instrument can do. We understand how to design a panel. Let's think about different controls that we need for our experiments. So as you're thinking about controls, you may need to have routine controls that are done daily or for each experiment. You may need to have just initial controls when just setting up your staining panel, or you may have experiment-specific controls that are maybe very specific for your antibodies or your cells, or something that's very specific for your protein-to-protein interactions. If we think about it, your QC control of your instrumentation, like our PMT calibration from our max quant, that is a routine control, and we're doing that every day we run our cytometer. Other routine controls are always unstained cells. You always want to run your unstained cells so we can verify our forward scatter and our side scatter so we can display our cells in our plot. And we want to always look at our single stains. And so we want to look at our single stain sample so we can compare that to our fully stained sample. And those single stains are also helpful for calculating manual compensation or automated compensation. And then there are very specific initial or experiment-specific controls, and one of them that I like to talk about is an FMO, or fluorescence -1. FMOs are very helpful to help you determine where to set your gates. And this is important as you start building bigger panels. When you have compensation, which is correcting for the spillover, you're going to get data spread. And so we see with little compensation, we see our signal here. And when we have a panel with lots of compensation, some of our data can spread out. And when we have an FMO in this case, this FMO does not have APC in it. I can draw a gate and I can see where my negative is and then my dynamic range for when I do add APC. But when I have a large overlap, an FMO allows me to truly determine and go, this should be where my negative region is, or where I should draw my gate for seeing the true signal of APC when I add it to the fully stained sample. All right, last feature, proper single cell suspension. And this is really critical for a flow cytometry experiment. It is so critical, I cannot do it justice in this particular webinar for you. And so with that, it has its own webinar of itself because there's many factors to go over for proper single cell suspension. So look on Miltenyi's website for other single cell suspension or sample prep for flow cytometry because you want to have good quality cells going into your cytometer. I will move into now workflow standardization and how you can standardize your flow cytometry experiments. Now, if you're new to panel design, don't worry because Miltenyi has got pre-designed and validated panels. And we've tested these panels so much that we know they are reliable and we're going to give you reproducible results. And we've labeled these MBTPs for Miltenyi Biotec tested panels, and we have a bunch of tested panels on our website. Speaking of reagents, you want to use good quality, high quality reagents to give you standardized reproducible results. Miltenyi has reaffinity antibodies, our Vio and our VioBright dyes. We get lot-to-lot consistency, and you're going to get bright, sensitive, and consistent signals. And for standardization, our instrumentation itself, our MaxQuant 16, you can have the MaxQuant 16 do your multi-sampling and cell labeling for you for consistency. And lastly, once you acquire your data, now you need to analyze it. Where do I draw my gates? What if I have those gates that are really close to the other populations? Is that gate going to be set correctly? We have express modes that are algorithm-based data analysis. So depending on where the cell populations are, the software will auto-adjust the gates and draw those gates for you. Let's take a look at a few of these. So as I mentioned, if you go to our website, miltenyibiotec.com, and at the top of the website, if you select applications, and then go down and go to all protocols, as I'm outlining here. When you go to all protocols, and you can again see applications, all protocols, you can see a list with step-by-step application protocols with our flow cytometry antibody panels. All of the MBTPs will be listed, and they have different categories and different cell types that we've used these on. I'm going to select this first one, this MBTP27, which is immunophenotyping of B cells from human buffy coat. When I click on MBTP27, I have the option to print the protocol. We can take a look at the protocol. So when I click protocol, I can see my staining protocol and exactly how to stain the cells, what centrifugation speed, and how to resuspend your cells. If we select up into the materials, you'll see the exact list of antibody and different markers that we've selected for this panel. The different clones that were used, these are my favorite reaffinity antibodies. What the purpose is, what that marker is used for, if you're new to a particular experiment or cell type. And you can see the fluorochrome that we have selected that goes with that particular marker, as well as the detection channel that will be used. You will also see gating strategy for how we've analyzed our data. So again, if you are new to flow cytometry, I highly recommend going to miltenyibiotec.com and checking out and seeing if we have already tested a panel that could be a good starting point for one of your new experiments. I also mentioned our antibodies, and I love recommending our reaffinity recombinant antibodies. How are these recombinant antibodies made? We use a human IgG backbone. So most antibodies have this Y-shaped structure. So this is the backbone. And where it's specific for each of the binding sites is on the outer end here. So we're using an example of a mouse IgG. So we take the specificity of our antibody bound to a human IgG backbone, and we have put it into a sequence and we culture it. And so we get the same consistent antibody production each time, and we have consistent lot-to-lot generation. Now, the nice thing about this recombinant antibody is this Fc portion is mutated, so there's no need for an Fc block. If you want to have isotype controls, that is another experimental type that I did not talk about in my previous slides, but a lot of people like to use isotype controls. which determines specificity of is this antibody binding to the antibody itself or the specificity of the endpoint of the antibody. If you want an isotype control, you only need to use one because the universal backbone only requires you to have one isotype control. Highly recommend for standardization, check out our reaffinity antibodies. They are excellent to use. Now, how can I have my cytometer do some auto labeling for me for consistency? Well, this is our robotic needle arm. You can see a close-up up here. And with our MaxQuant 16, we can show you how the robotic needle arm can come out and take your reagent of interest, add it to your cells, mix the cells up, and then aspirate a certain specific amount for acquisition of your cytometer. This auto labeling with our robotic needle arm is excellent for very consistent results well to well in a 96 well format such as I'm showing you here. Lastly, we have our express modes, which is those algorithm-based gate placements. And this is showing an example of our 8-color immunophenotyping express mode. Miltenyi has an 8-color immunophenotyping express cocktail where we have all of those antibodies put together. It's a quick 10-minute incubation. You put it on your cytometer, our MaxQuant. We have the MaxQuant express modes, and we can see that the software has put in these gates based on the cell populations, defined all of the different populations, and we have a list of all of our specific types of our B cells, T cells, and NK cells. If you're using or looking at basic immunophenotyping, I highly recommend this 8-color cocktail kit to use for staining, as well as our express mode. So there's no guessing as to where you should place your gates. And with that, I thank you for your attention for today's seminar. Please visit our website, www.miltenyibiotec.com, to help you standardize your flow cytometry and get you excellent specific results that are very reproducible. Thank you for your attention.